Okay, so we are going to get started. I want to welcome everyone to a highly anticipated event in our Provost Distinguished Speaker Series for this academic year. I'm Carl Lejwe, a professor in the Department of Psychological Sciences and your Provost. The Provost Distinguished Speaker Series is an annual set of events where we invite our recently appointed Board of Trustees, distinguished professors, and endowed chairs to share their scholarship with us. We're really pleased to share this opportunity to bring our community together in appreciation and celebration of the exceptional work we do here at UConn. I will say, in the 18 months that I've been here, probably the thing I have heard the most is we don't do enough to tell our academic story. We don't do enough to tell the UConn story. And I think events like this are a really important part of our push to do this more and more, because when we think about the specialness of that story, it's hard to think of more exciting research and someone would want to celebrate more than Dr. Marja Hurley, professor of medicine and orthopedic surgery at UConn School of Medicine. She'll be sharing her scholarship with us today. She's one of our newly appointed BOT distinguished professors, and her talk today is titled Osteoarthritis of Mice and Humans from Bench to Bedside. The BOT Distinguished Professorship is the highest honor that the university bestows on faculty who have demonstrated excellence in teaching, research, and service, and in doing so elevates the reputation of our university. One of the best parts of being provost is getting to recognize our top scholars with this award, as well as to feature their scholarship in this forum as an opportunity for our community to learn about it, ask questions, and engage. Dr. Hurley has an extensive and impressive list of accomplishments, which I will leave to her dean, Bruce Liang, the dean of the School of Medicine. But before I do that, I always like to say a little bit about the dean who'll be introducing our distinguished professor. Now, it says a lot about the School of Medicine. I've already had a chance to do this for Bruce very recently, so instead of going on and repeating myself, I'll just say one thing. What is always really important in an administrator is that what they say and what they do is the same thing. And whether that comes to where they invest resources and where they say their vision for a school is, or where they say their values are and the decisions they make. What I really appreciate about Bruce is he is someone who does what he says. And where I really want to acknowledge that is in the support and development of research at our university. He is someone first who has shown that he understands what it means to do this because he's done it himself. And he's someone who is a tireless advocate for his faculty and staff and student researchers, really pushing the university to provide as much support as possible, providing mentorship, and particularly providing support, because we all know it's easy to talk about expanding research, but it's incredibly hard to do. And I really appreciate who Bruce is at this university and what it means to have a leader who not only says research is important, but shows it in everything he does. So with that, I'll hand it to Bruce.
Well, thank you very much, Carl, for those kind and encouraging words. And it really gives me great pleasure to introduce Dr. Hurley. First of all, congratulations, Dr. Hurley, Marja, once again, for your BOT distinguished professorship. And it really is a great honor for the School of Medicine for you to get bestowed this highest recognition. And if I were to tell you about Dr. Hurley's accolades and recognition, I'll be here till tomorrow morning. So I'm not going to do that. Obviously, you want to hear from Dr. Hurley. And I will just mention a few highlights of Dr. Hurley's long and distinguished career. Dr. Hurley was the first University of Connecticut Martin Luther King Award for Achievement in Science, an honor she earned in 1990. And she went on and earned a number of additional awards and accolades, including in 2010, winning the Connecticut Council Women of Innovation and Leadership Award, and the Ray and Carol Niak Award in 2011. And she is a member of the Connecticut Academy of Science and Engineering and a trustee of the Connecticut Science Center. Professor Hurley recently accepted the prestigious invitation from NIH Director Francis Collins to serve as a new member of its National Institute of Dental and Craniofacial Research Board of Scientific Counselors, a real tribute to Marja's accomplishments and recognition, and bring additional reputation and visibility to the School of Medicine and therefore to the University of Connecticut. Marja is widely recognized as a leading authority in the world on fibroblast growth factors in osteobiology. And her pioneering work has shown how FGF2 protects against development of osteoporosis and osteoarthritis, a very common, or two very common conditions affecting a lot of us, unfortunately. Her study has advanced our understanding of this very important molecule. She has published over 100 scientific papers and reviews, delivered many invited presentations. Her work has been supported by more than 10 million in grants from the NIH. And another very important accomplishment for us is that she has had a long history of dedicated service at both the college and university level, including the fact that she has served for 34 years as Associate Dean and Founding Director of the Aetna Health Professions Partnership Initiative at UConn Health, where she focused on developing STEM programs for middle, high school, and college students in order to diversify the health professions, for which she garnered more than $14 million in extramural grants. And so once again, you really want to hear from Dr. Hurley, but this is just a glimpse of her accomplishments. So allow me to hand the podium over to Dr. Hurley. Thank you very much.
Thank you, Dean Liang. Vice Provost Bradford, Provost Leger, I would like to thank you for the invitation to participate as a speaker. And I'm deeply honored to have been selected as a Board of Trustees Distinguished Professor. I would like to thank my nominators, Dr. Lenworth Ellis in the School of Medicine and Dr. Lisa Kuhn in the School of Dental Medicine, as well as my colleagues at the national and international level who wrote letters to support my nomination. As some of you are aware, I received both my undergraduate and graduate medical education at the University of Connecticut. And at this time, I would like to acknowledge two of my mentors during my undergraduate years at Storrs. And the first individual is Dr. Fred Adams. Dr. Adams was the Vice President for Student Affairs, and he also was instrumental in founding the School of Allied Health and served as its dean. His commitment to health professions diversity and his support of my initiatives as a medical student at UConn influenced my work in developing the health career opportunity programs at UConn Health. Unfortunately, he passed away too early in life. The second individual that I would like to recognize is Professor Heinz Hermann. He was my cell and molecular biology professor in a class of 100 or more students. And somehow we connected, and He remained a mentor and supporter of mine until he passed away at the age of 98. For the students in the audience, I want to point out that Dr. Hermann, along with 4 other distinguished researchers, founded the American Society for Cell Biology in 1960. And to honor his achievement, the ASCB presents the Dr. Heinz Hermann Symposium every year at its annual meeting. I also believe that there is another symposium that's held annually at stores in recognition of Dr. Hermann. The other individual that I want to mention, Tom Deutschmann, who has been a mentor and scientific collaborator to this day. And it was really very interesting that Tom obtained his PhD at the University of Connecticut, and his thesis advisor was Professor Hermann. Tom and I met almost 20 years after he was at UConn, and Heinz got a kick out of knowing that we were both collaborating together. Dr. Deutschmann did his postdoctoral fellowship at the Max Planck Institute and also at the University of Wisconsin in gene targeting under Oliver Smithies. And in 2007, the Nobel Prize in Physiology or Medicine was awarded to Mario Capecci, Martin Evans, and Oliver Smithies for their discoveries of principles for introducing specific gene modification in mice by the use of embryonic stem cell. And during his Nobel lecture, Oliver Smithies noted that Tom Deutschmann did the seminal experiment in the lab where he corrected the mutant form of HPRT gene that had been generated in stem cells in Martin Evans' lab. And that paper was published in Nature in 1987. And for those of you who may recognize this gene, this is the gene that is abnormal in the human disorder Lesch-Nyhan syndrome. Now, luckily for me, Tom also knocked out another gene, and that was the FGF2 gene in mice. And this was a paper that he published in Nature Medicine in 1998. And I was fortunate to obtain the mice from Tom. And quite frankly, that mouse resulted in my obtaining my first NIH R01. Now, the fibroblast growth factor family consists of 22 ligands and 4 receptors, and although there's an FGF23, which we'll get to later on. But I've spent the last 20 plus years studying FGF2 gene, and this is an outline of what I would like to discuss with you today. The FGF2 gene encodes for these very unusual protein isoforms. And we made transgenic mice harboring these isoforms, which we'll talk about a little bit more. We also, I'd like to discuss how high molecular weight FGF2 may have some role in X-linked hypophosphatemia and osteoarthritis. I'll talk a little bit about the FGF receptors in osteoarthritis and the gap in knowledge of the potential role of FGF2 and FGF receptors in osteoarthritis of XLH, provide you with some studies that we've done in the laboratory, and then just some summary conclusion and future directions in terms of potential clinical translational studies. Now, the FGF2 gene, which I'm showing here, encodes for these unusual high molecular protein isoforms that have nuclear localization sequences. And they appear to act like transcription factors or bind to DNA directly or through binding with other factors in the nucleus. In humans, there are 4 high molecular weight isoform, and there's a low molecular weight 18-kD isoform that is exported from the cells because it does not have a signal peptide, but it functions in an autocrine and paracrine manner. Now, many years ago, I did my one and only 6-month sabbatical and never took another one. My patients were upset that I was gone for 6 months, but I brought back the cDNAs for these different isoforms. And we utilized a fragment of the Col 3.6 promoter that was made by Professor David Rowe here at UConn. And we used that to drive the expression of green fluorescent protein, which we wanted to be able to track where the gene was being expressed. And so that was our vector mouse, as I call it. This was our control mouse. And then we used the promoter fragment to drive the cDNA for the high molecular weight isoforms, as well as GFP sapphire. And what we observed was that the mice that I will call going forward the high molecular weight TG mice, that they were much smaller. And this is vertebrae from the vector mice and the high molecular TG mice. And you don't have to know much about micro-CT to see that compared with the vector, you have all of these nice trabeculae. And here in the high molecular weight TG mice, you have a lot of holes. And so these mice had decreased bone mineral density, they had dwarfism, they had osteomalacia rickets, they had hypophosphatemia, and they had increased FGF23, even though I just told you that there are only 22 ligands. The reason for that is the gene for FGF15 and 19 are the same mouse and human homologs. Now, for the students in the audience, go and visit your mice in the animal care facility. And one of the postdocs came back, and we do aging studies in our lab. And they indicated that some of the mice were having some difficulty ambulating. So we decided to just obtain some x-rays of mice at 2 months, 8 months, and 18 months of age. And this work was the work of my former PhD student, Patience Meobert. And what we saw was, even at 2 months of age, we saw that the blue arrow, there was some thinning of the femur. This is the femur, this is the tibia. Give you a little anatomy as I go along. And there was also some flattening of the tibial plateau when you compare the nice rounded end here. And and comparing mice with age, we see that there was further thinning of the subchondral bone, and also more evidence of an early osteophyte, which is an abnormal bony protrusion, which one should not see in the knee joint. And then you don't have to know much about anatomy to see that, boy, this joint looks terrible. It really is destroyed. What we can see is, with the green arrow, shows that there's narrowing of the patellar femur junction. The space is gone. There is more evidence of sclerosis of the bone, and this osteophyte that I spoke about appears to be more pronounced. So these phenotypes reminded us of a clinical disorder, X-linked hypophosphatemic rickets. And XLH affects about 1 in 20,000 people, and is caused by a mutation in the PEX gene, resulting in abnormal cleavage of the hormone FGF23, which leads to phosphate wasting from the kidneys. And among the morbidities and complications is that these individuals develop osteoarthritis that can be severe enough to warrant joint replacement. And so new therapies are needed for OA in XLH subjects. And this is just individuals with XLH, and almost all of these individuals will have osteoarthritis, and some may need to have their joints replaced. And this is just, again, similar to the mice. You can see that in the humans, the red arrow shows that there's loss of the joint space compared with the blue arrow showing the nice spacing of the femur and the tibia. And there's articular cartilage here, which allows us to bend our knees, and we don't have two bones rubbing against each other. Now, I indicated that our mice phenocopied XLH, but we also wanted to determine whether or not this was just an observation, or was there any evidence for this? So there's a mouse homologue of XLH called the hip mouse, and these mice, similar to humans, develop osteoarthropathy. So we examined the hip mouse, and we found that they expressed increased FGF23, and these are osteocytes. And also there was a marked increase in FGF2 in these mice, which no one had ever reported before. And we see that the yellow, that there's overlapping. So within the osteocytes, we see both. So the research question is, do FGF2 isoforms contribute to OA in high molecular TG mice that phenocopy osteoarthritis of XLH? And again, just a little bit more anatomy for you. So here again is the femur, and here is the tibia. And we have this nice blue cartilage joining between the two bones. And so I'm just showing you a little blow up of this area of the articular cartilage, the subchondral bone, and also the meniscus. And just very briefly, what happens is the joint is an avascular structure, and the chondrocytes within that space usually remain undifferentiated. And during osteoarthritis, they begin to differentiate. They become what we call hypertrophic, and then they begin to become calcified. And if you think about that, you could see how this could, cause patients to have, significant pain and also a loss of range of motion. And these abnormal differentiated chondrocytes also produce a lot of inflammatory cytokines and also enzymes that will break down the tissue, such as MMP13, which I'll talk about. And so then you have a thinning of the articular cartilage. The subchondral bone is also involved, and there's increased bone remodeling, and there is sclerosis of the bone. Now, the literature has indicated that there's a role for FGF2 and FGF receptors in osteoarthritis. And the dogma is that both in mice and humans is that FGF2, if it binds to the FGF receptor 1, it will activate a downstream signaling pathway, such as a RAS-MEP kinase pathway, resulting in increase in the transcription factor RUNX2, which will then be translocated to the nucleus and will affect genes that will contribute to matrix degradation and to destruction of the cartilage. However, if FGF2 binds to the FGF receptor 3, then it is anabolic or protective. But what was not known until our studies was whether or not the different isoforms of FGF2 may have different roles that had not been considered. And so, there are no reports on the effects on inhibition of FGF receptor signaling on OA in hip mice or humans with XLH. And so our hypothesis was aberrant FGF receptor signaling contributes to the OA observed in high molecular TG mice. And we wanted to examine whether this OA can be rescued with an FGF receptor inhibitor, BGJ398. Now, this inhibitor is actually FDA approved for the treatment of certain malignancies. And the next question we wanted to assess was whether FGF receptor blockade modulates increased canonical Wnt signaling observed in osteoarthritis knees of the high molecular weight TG mice. So very briefly, what we did was we had two questions. We wanted to know if we started treating the mice with this inhibitor just when they were weaned, and we weaned the mice at about 21 days of age, could we prevent the development of the osteoarthritis? So the first set of data that I will show you reflects our treating these mice for 12 weeks with either the vehicle or BGJ. And this shows the X-rays. You can see here that there's narrowing of the joint space, again, the femur and the tibia, when you compare with this nice space that you can see between the two, between the two bones. The other observation is that if you look at the surface of the bone, the articular surface is eroded. And what we see with treatment with BGJ after those 12 weeks is that you can now see that there seems to be some rescue of the spacing here, and you also see that this pitting that you saw here is no longer present. Really quite beautiful. And this is just a lateral view. Again, you can see just a big hole here in the femur and in the tibia, the subchondral bone, compared with the vector mice. And you can see that the treatment with BGJ rescues this. And in the interest of time, I'm not going to go through all of this, but the bottom line is that we can see that there is a reduction in the bone volume, trabecular volume, and there is an increase with the BGJ treatment. Now, we also, I indicated to you that part of the problem is that we have a loss of the articular cartilage, and proteoglycan makes up quite a bit of that cartilage, and you can stain for that using a safranin O. And if you look at the high molecular weight vehicle treated, you can see that there is a marked decrease in the amount of safranin O staining compared with the vector vehicle. And you can also see that in the animals that were treated with BGJ, that there is some rescue of an increase in the proteoglycan. Another abnormality which you should not see in the normal joint is you should not see alkaline phosphatase, which is a marker of calcified cartilage or bone. And you can see that that's very quite prominent here, and that this is rescued with BGJ treatment. We can measure the articular cartilage thickness, and we can see that there was a significant reduction and a significant increase following treatment. I mentioned that there are changes in enzymes, and there's also a change in the type 10 collagen, which is a marker of the more differentiated chondrocytes. And you can see that's increased, and there's a partial reduction with BGJ. The degrading enzyme, MMP13, was significantly increased and was also reduced by BGJ. And just to show that this was not just changes in messenger RNA, we looked at the level of the protein, and you can see compared with the control or normal mice that there was a marked increase in the protein for MMP13, and this was nicely rescued with BGJ. And we quantitate this by measuring the amount of positive staining, significantly increased, significantly reduced by BGJ. So the next question that we wanted to ask was, does FGF2 and FGF receptor modulation of Wnt signaling contribute to OA in the high molecular TG mice? And I will tell you that, the etiology of osteoarthritis is not known. There are lots of studies suggesting that there's increased Wnt signaling, there's increase in bone morphogenetic protein, there's increase in the FGF2, there's increase in inflammatory. So in other words, we're just focused on one potential pathway. And again, let me try to take this through this somewhat very busy slide. So the bottom line of this is that there are molecules called inhibitors of the Wnt signaling pathway. And if they're increased, they shut down the ability of Wnt to signal, and you have the most important factor, beta-catenin, becomes ubiquitinated and degraded and is not able to translocate to the nucleus. What we believe is going on is that high molecular weight FGF2 increases FGF23. We've shown that this occurs, that high molecular weight FGF2 transcriptionally regulates FGF23. FGF23 is an endocrine hormone, which means that it gets out of the cell, it circulates around the body, it goes to the kidney and other tissues. And we think that FGF23 binds to the FGF receptor and in some manner activates the Wnt signaling pathway so that you have an increase in an important protein axin. You have binding of Wnt ligands to their receptors, and this results in the dissociation of these proteins that are that would ordinarily degrade beta-catenin. Beta-catenin can then go to the nucleus and binds to different genes and affect gene transcription. And in this case, we're interested in its effect on MMP13, type 10 collagen, VEGF, and FGF23 itself. So very quickly, we determined whether or not there was increase in FGF23 itself in the articular cartilage, and we show that. Again, we were the first to demonstrate this. There's FGF23 is ordinarily increased in the subchondral bone because it's made by osteocytes. And so when we did our quantitation, we see that there was a significant increase in FGF23 in the articular cartilage. This was reduced by BJJ treatment. There's really no effect of the FGF23 in the subchondral bone. There was also an increase in serum FGF23, and there was a partial decrease with the BJJ treatment. The only when you treat with a drug, and if you have normal controls, you're always concerned about off-target effects. And so that's one of the reasons we treated the normal animals with the BJJ as well. And the only observation that we saw was that there was a slight increase in serum FGF23, which could be of concern because it controls phosphate, but we did not see an elevation in the serum phosphate. Now we looked at the FGF receptor. I already told you that that is the receptor. FGF receptor 1 appears to be important in osteoarthritis. And we see again a nice increase in the articular cartilage and a reduction by BJJ. FGF receptor 3, however, we did not see much in the way of a change in FGF receptor 3, which is supposed to be protective. And when we quantitate, there was a significant increase in the phosphorylated FGF receptor 1 and a significant reduction by BJJ. Getting more into the Wnt molecules, we saw that a SOS, which is the Wnt inhibitor, was reduced in the high molecular weight vehicle and was increased in the with BJJ treatment, and I'll come back to the significance of that later on. Axin 2 was markedly increased and was reduced by BJJ. So what we see is that there was a reduction of SOS in the subchondral bone, and this was increased by BJJ. Contrast, Axin 2 was markedly increased and was reduced by BJJ. And just a couple of other components of the Wnt pathway. There was a marked increase in Wnt7b, one of the ligands, and also a corresponding increase in the receptor to which the Wnt ligand would bind. And BJJ treatment reduced both of those. And then quantitation, a significant increase in Wnt7b in the vehicle high molecular weight TG mice, and this was reduced by BJJ treatment. And the phosphorylated form of the receptor was LRP5 was significantly increased and reduced by BJJ treatment. And then we also looked at the active beta-catenin, which I indicated the transcription factor that's going to go to the nucleus and cause all of the problems. And you can see that there was a marked increase in beta-catenin, and this was reduced by BJJ treatment. And again, quantitation of this, a significant increase in staining for beta-catenin and a significant reduction by BJJ. So now let me go back to what we think is happening. So we think the FGF receptor inhibitor, it will interfere with the binding of the interaction of FGF23 and its receptor. And when this occurs, you will now have an increase in the Wnt inhibitor, which will essentially shut down the increased Wnt signaling to prevent beta-catenin from going to the nucleus. So in summary, increased FGF2, FGF23, and FGF receptor 1 expression in articular cartilage of high molecular TG mice compared to vector TG mice. The FGF receptor inhibitor BGJ partially rescued the OA phenotype in these mice, and BGJ398 blocked the increased canonical Wnt signaling in knee joints of high molecular TG mice. So we speculate that high molecular FGF2 contributes to OA in mice via altered FGF23, FGF receptor, and Wnt signaling. So what is the clinical significance of our studies in mice? So this is a potential therapeutic approach that partially rescued OA in vivo in mice using an FDA-approved FGF receptor tyrosine kinase inhibitor. These findings are novel since there are currently no published disease-modifying drugs to treat the degenerative joint disease of XLH subjects. I should point out that approximately 2 to 3 years ago, burosumab, which is an FGF23 blocking antibody, was approved by the FDA, but there's no data as yet about its efficacy in osteoarthritis of XLH. And this is just a piece of data which I wanted to share with you. We wanted to ask the question: if we treated the mice after they had developed osteoarthritis, so the older mice, could we rescue or improve the phenotype? And I must confess that we were in the middle of these studies during COVID, and we had to, stop the experiment after 6 weeks because all the labs were being shut down. But we found some interesting phenotype. We found that the high molecular weight TG mice, again, this loss of trabeculae and loss of the spacing. And you can see that even in these mice that are now, we treated them for 6 weeks. So we started when they were 8 months, so they're 9.5 months old. There was, a significant rescue of the phenotype. So the university has filed a patent application for potential use of FGF receptor blocking to inhibit osteoarthritis progression in the high molecular weight fibroblast growth factor 2 transgenic mouse model that phenocopies human XLH. So obviously, potential clinical trials using BGJ receptor that is already approved for treatment of malignancy is something that we should consider. And obviously, these are discussions and potential studies that would have to be done with my rheumatology colleagues. I just wanted to point out some individuals in the laboratory. Dr. Liping Zhao, she was a postdoctoral fellow with me. She's now an assistant professor and conducted a lot of the studies. I've already mentioned my former graduate student, Patience Miabert, who did some of the studies in the younger mice. Danielle Williams, who's now a second-year medical student here at UConn, also contributed to the work. And there are 3 graduate students in the lab. I should say that this picture is pre-COVID, so nobody's wearing a mask. And we don't have pictures of Kai Clark, Michael Tassifo, or Christine Valera, who are the graduate students in the lab at the present time. I wanted to just acknowledge the UConn Center for Mouse Genome Modification, who assisted and helped us to make that mouse many, many years ago. And the beautiful images from the micro-CT, I wanted to acknowledge the micro-CT imaging facility at UConn Health. You know, part of the gratification is the trainees. And over the years, I've had students who were, I served as either their major or associate thesis advisor for the PhD, for the master's. Other students have rotated through the lab who, I've also supervised a number of postdoctoral fellows, visiting scientists, residents, fellows, medical students, dental students, and college students. And none of this would be possible without the support of NIH. But I also wanted to acknowledge this this first grant, and just want to give a shout out to everyone who has an R01 or other grants. I was able to start my research career because Dr. Lawrence Royce, who was the head of the Division of Endocrinology and another of my mentor, he had a program project grant, and we applied for a minority supplement, which I used for 3 years to obtain preliminary data for my first R01, which was funded on the first try. Trust me, that doesn't happen too often after that. You have to keep resubmitting. But I just think that this is something that we should utilize to support undergraduate students, graduate students, and obviously as a junior faculty, it provided support for me so that I could obtain the preliminary data for my grant. And then for a clinician, you know, for me, this was just an incredible gratification. This is our Bible, the endocrinology, and this was the 100th anniversary of the endocrinology, and they asked if they could put the bones of our mice on the cover. Of course, I said yes. Thank you. And so this is the end of my talk. Thank you very much.
Thank you very much. We so appreciate it. I just want to say to all colleagues joining us, the floor is now open. You do not want to leave the Q&A to a playwright. There's absolutely no way I'm going to have any kind of in-depth conversation with Marja about this. Was so to the information about the clinical significance, because I could follow that, and the BJJ treatment, which sounds really positive. And I'm always curious about, you know, clinically what is possible. And I think under.
Oh dear, I hope I'm not having trouble with our. Ooh. Yes.
Okay, all right. Well, I just want to open. I have a question, pretty naive, but I don't want to even be there. So I'm hoping that our colleagues won't mind jumping in and asking you some questions. And folks, you can unmute yourself and jump right in. And folks, hear me? I'm connected. Can I get a thumbs up, Marja, if you can hear me?
Yes.
Okay, great, 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 great. The floor is open, but I am going to ask a really naive question, if I may. And so I'm curious about the clinic of the work that you are doing. Sounds like there's a part of this research is in some trials where it's up for approval. I'm research. I knew that the cartilage prevalent issue, but I didn't know how until I read, you know, some of your material. Love if you can talk about what the next bit are and how this might affect folks with this issue.
So one of the things that I want to make clear about this study is, you know, there are many causes of osteoarthritis, and I want to restrict my my comments to this genetic disorder, which can be associated with osteoarthritis. I don't want to give anyone the impression that you know, this could be a treatment for osteoarthritis in general, even though the genetic osteoarthritis that we see in XLH looks very much like osteoarthritis that is everyone gets. So whether or not it has any application above and beyond the individuals with this disorder, I think that would be something that we would want to explore at you know, with further studies. Yeah.
Fantastic, fantastic. Let me invite our participants to mute and join in the conversation.
Well, I hope it was clear. No questions.
Hello, can you all hear me? I'm thinking either I have a problem or the network has a problem.
Well, I've seen some congratulations in the chat, but no questions.
Hear me? It's Michael. Marja, can you hear me?
Yes.
Okay. I'm just curious, and I said forgive my naivete in my questions, but I'm curious, what is the next frontier? Like, what is the next space to explore research?
So as I indicated, I think, you know, the university has filed this patent, and I think that now we will see whether or not you know, how we move forward with this. I think that obviously discussions with my colleagues who are treating patients with XLH, or whether obviously before you start treating patients, there are lots of other studies that would need to be done. But the thing is that this drug is already approved for certain malignancies, and we're talking about a state where there is overexpression of or hyperactivity of the receptor, and we know that not only the receptor, but at least these isoforms of FGF2 appear to be increased, at least in the mouse model of XLH, and I've already contacted some collaborators to see if we can actually confirm that these isoforms are also increased in humans with XLH. We've shown it in the mouse model, but we still need to do that.
My goodness, my goodness. Thank you for that. I appreciate it. I appreciate it. So I just, this is to our enemies out there in the world. It's the naive players like myself who have questions. I hope you didn't mind it.
Oh no, not at all. Not at all.
I appreciate it.
Hey.
That's me and my own echo. Marja, I just want to say thank you so much. Congratulations again on the designated as distinguished professor. More than well deserved. We appreciate you coming in today and having this conversation with us all. It's important work. Appreciate and congratulate you on all your accolades and just an all around thank you. You know, the level of support you've given me personally while here at the university is something that I really appreciate, and I try to follow your lead at every turn. So I just want to say thank you, and I appreciate you.
Thank you very much.
All right, all right. And everyone on the call, thank you so much for joining us today. We really appreciate it. And I'm sure Marja does as well. And everybody have a really wonderful night. All right.
Thank you.
Take care all.
Bye-bye.
Thank you. Bye-bye.